Unit 313, we do practice. Solution X has an absorption spectrum shown on the graph below. What would be an appropriate wavelength to gather the absorption data? So typically when given the absorption spectrum for a particular substance, we're looking for the highest peak or the peak that is closest to 1. And in this case, our highest peak is right here. So when we drop this down, it's right on this line, and our x-axis is our wavelength in nanometers. So we've got 600 here, 700 here, and I've got 5 lines in between. So each one of those is going to be 20. So when I drop this down, this point right here would be 620 nanometers. So an appropriate wavelength to gather our absorption data for this particular solution X would be 620 nanometers. So part B asks us what is the color of the light absorbed. So it's 620 nanometers. That's going to fall right here in the orange section, which goes from 635 to 590 nanometers. So our 620 is going to be right in there. So the color of light absorbed is going to be an orange light at that particular wavelength of 620 nanometers. Part C asks us to predict the color of the solution. And remember, whatever light is being absorbed, its complementary color based on the color wheel is what we're going to see, or what is going to be the color of the solution. So if we're absorbing orange light, then our complementary color is blue, so our solution should appear blue. Finally, for part D, they tell us that a solution with an unknown amount of the same solute X was prepared and graphed as shown. How is the new solution of an unknown concentration different from the first one, and how is it the same? So taking a look at our graph, our new solution is in red, right? This is our absorbance of our unknown solution, and then blue is our absorbance of our original of solution X. So they want to know how these two solutions are different and the same. So the first thing we notice is that their peak absorbance is the same. It's at 620 nanometers. So what this means is that if their peak absorbance is still the same, then they're both still absorbing that orange color wavelength. So the color of these two solutions must be the same. So that's how they're the same. However, they're different in overall absorbance. So we notice the blue is absorbing a lot more than our red solution. So how they're different is that if red is absorbing less, then it must be less concentrated than our solution represented by the blue line. So our red line solution, our unknown solution, actually is less concentrated than our original. And we can actually go a step further because we notice on our blue, its peak absorbance is at 0.8, and red, its peak absorbance is at 0.4. So our unknown solution actually has half the concentration. And it told us right here that our original solution is a 1 molar solution. So our unknown solution with an unknown concentration, that concentration actually must be 0.5 molar. So that is how they are the same and different.